Bismillah ar-Rahman ar-Rahim. This clinical scenario is based on the importance of phospholipids in lung expansion. So, in this clinical scenario, the cues to diagnosis include steroid injections, fetal lung maturation, preterm delivery, and respiratory complications. All these point towards the acute respiratory distress syndrome or the hyaline membrane disease. So, these are the possible questions which may be asked. Surface tension is created due to cohesive intermolecular forces within water molecules with hydrogen bond formations and the tendency of water molecules to assume a symmetric arrangement on the surface, along with their tendency to assume a low energy spherical state as seen in falling raindrops. Although the function of fetal breathing movements is not entirely clear, they can be observed starting at 20 to 21 weeks of development. Fetal breathing movements involve muscle contractions that cause the inhalation of amniotic fluid and exhalation of the same fluid with pulmonary surfactant and mucus. Prior to birth, the lungs are filled with amniotic fluid, mucus, and surfactant. As the fetus is squeezed through the birth canal, the fetal thoracic cavity is compressed, expelling much of this fluid. Some fluid remains, however, but is rapidly absorbed by the body shortly after birth. The first inhalation occurs within 10 seconds after birth and not only serves as the first inspiration but also acts to inflate the lungs. Pulmonary surfactant is critical for inflation to occur as it reduces the surface tension of the alveoli. Preterm birth around 26 weeks frequently results in severe respiratory distress. Although with current medical advancements, some babies may survive. Prior to 26 weeks, sufficient pulmonary surfactant is not produced, and the surfaces for gas exchange have not formed adequately. Therefore, survival is very low. In the early stages of gestation, the water in amniotic fluid is derived mostly from the maternal serum. Later on, during gestation, as the amniotic fluid expands, fetal urine becomes the largest source through the amniotic fluid. Lung secretions, gastrointestinal secretions, and excretions from the placental services contribute to the composition of amniotic fluid as well. However, lung secretions alone make up as much as 1/3 amniotic fluid. Without surfactant, fetal lungs cannot overcome the surface tension forces caused by water lining the lungs, and this surface tension leads to collapse of the alveoli. 10 times the normal force will be required for lung expansion in case of surfactant deficiency at birth. Not only that, even if the lungs are expanded artificially, for example by endotracheal intubation, the water lining the alveoli in absence of surfactant will lead to their collapse, as shown here. An important biochemical basis for the fetus being able to survive outside the uterus after roughly the 24th week is that the type 2 pneumocytes start producing small quantities of lecithin. Thus, the ratio of lecithin to sphingomyelin in the amniotic fluid increases with increase in fetal age. So that, here you can see the lung has collapsed. The alveolus, and it is due to the deficiency of the surfactant. Pulmonary surfactant coats the alveolar space of lung to reduce the surface tension and prevent collapse of the alveoli during breathing. Pulmonary surfactant is composed of lipids, 90% of its weight, Surfactant-associated proteins form 10% of the weight. The most abundant lipid with surface tension lowering properties is dipalmitoyl phosphatidylcholine or DPPC, 60 to 70% of the weight. DPPC is necessary to achieve the surface tension of near zero during the compression and expiration. Surfactant-specific proteins include SPA, SPB, and SPC and SPD. SPA and SPD are hydrophilic proteins with no surface properties, whereas SPB and SPC are low molecular weight hydrophobic proteins which lower the surface tension and prevent the alveolar collapse at the end of the expiration cycles. Lecithin Sphingomyelin ratio serves as an index of lung maturity. A value of 2 or greater is evidence of maturity. It reflects the shift towards more lecithin synthesis after 32 weeks of gestation. Type 2 pneumocyte cells release both surfactant lipids and the hydrophobic proteins in response to a variety of stimuli through exocytosis. These are synthesized and stored in storage granules termed lamellar bodies and released upon appropriate stimuli by exocytosis. Once released into the alveolar space, the freshly secreted lamellar bodies help to establish the surfactant active lining layer. Cyclic compression and expansion during ventilation result in a fraction of spent surfactant that will largely be recycled. Thus, single constituents of surfactant may run through several cycles before being removed by alveolar macrophages and replaced by de novo synthesis. The status of fetal lung maturity is reflected in the concentrations of specific phospholipids and the number of surfactant containing particles or lamellar bodies in the amniotic fluid. The components of surfactant like DPPC, sphingomyelin, cholesterol, and hydrophobic proteins intercalate between water molecules and break the hydrogen bonds, leading to disruption of arrangement of water molecules and hydrogen bonding, thereby reducing the surface tension. So here we can see the proteins and cholesterol and phospholipids breaking the symmetric arrangement of the hydrogen bonds. Females with high risk pregnancies during which preterm birth is anticipated are administered steroid injections for fetal lung maturity. And fetal lung maturity is dependent on surfactant formation. After injection, the steroid hormones enter the cytoplasm by diffusing through the plasma membrane, encounter their receptor in the cytoplasm, and receptor steroid complex traverses the nuclear membrane, binds DNA, leading to enhanced transcription of genes which encode for the enzymes required for the synthesis of lecithin. So that's the steroid, and the steroid hormone complex has moved into the nucleus and leading to the transcription of the genes and synthesis of the enzymes. So, surface tension can lead to collapse of the lungs in preterm infants, and infants are not viable before 24 weeks because type 2 pneumocytes are not producing the surfactant. Steroids can lead to the formation of lecithin through gene transcription, and surfactant helps in lung expansion by breaking down the forces causing the surface tension, and fetal lung maturity may be determined by the lamellar body count or lecithin sphingomyelin ratio. and the normal lecithin sphingomyelin ratio is 2:1.